we'll let everyone try to take a seat. And thanks, there are some seats up in front. Get a bird's eye view. How is the audio? Folks in the back, Deb, good? Thanks, awesome. And visual okay back there? I might move around a little bit, so if you see me over here and you can't hear me, I'll have to go back over here. Terrific. Well, welcome everybody. This is kind of big shoes to fill after the plenary, but so excited that you're here and really eager to share with you our symposium today. We have some wonderful speakers, wonderful collaborators. I have personally been really engaged we've all been engaged, had a number of meetings, and you'll hear how this came to be over the next few minutes. We have Sina and Vin, Asia and Doug, and myself. I'm Susan Oliveira. I've been part of ICPE for a long, long time. I've been in the field over 25 years, and super excited to talk about something that I'm really passionate about, which is coding lists and the development of them and for use in pharmacoepidemiology and the how and the why. We have a back door, so folks might come in. First, I'd like to acknowledge two folks that were part of this but are not part of the symposium today. Ali Szynski and Deb Casso, very much part of the idea for this and very much part of the development of it. So thank you, Deb. Thank you, Ali. I see you in the audience. Terrific. So just to give you a little sense of where this came from, you know, these ideas for symposiums, they kind of percolate and generate. And all of us on the panel noted that coding lists, although they're a small piece of a study, they're a very important piece of a study. And what we acknowledged is that we felt as though we didn't get a lot of training in it. There isn't a lot of training in it. And there's a heterogeneity around how we develop these. And I know that in the audience, we have academics, we have industry, pharma, service providers, regulatory folks. And so across that spectrum, we have different uses of code lists and different types of implementation of them. And so that was the genesis of how we thought about this. And initially, when we put the abstract together, we had a very a tighter lane of how we were thinking about it. But immediately, as we all got together and met on multiple occasions, brainstorming around what would make this a great symposium, quickly we realized we had to talk about algorithm development, we had to talk about validation, we had to talk about dissemination, we had to talk about use cases, and also the effects of bias. So as you can imagine, this kind of exploded and bloomed. And so today we are going to be covering those areas. And we're going to be looking at it from a service provider, from a regulatory lens, an academic lens. And we're going to start out in laying some infrastructure. And we will build on that. And so what we'd like to do is to hold all questions until the end, because it is going to be very iterative as the speakers present their work. And we will start with Sina laying the foundation for code lists. And as I mentioned, the heterogeneity around how we develop these and the uses, right? It depends. If you're trying to do a quick feasibility or develop a hypothesis, you might do something more down and dirty. If you are doing it for a regulatory submission, it's going to be a different approach. And so we want to make sure that we highlight that, but also create some rigor. And the one goal today is really around raising awareness. We're not going to present a formal process. We're not going to say this is how to do it. But we want to raise awareness around the importance for coding lists and how those fit into the work that we do. And today we will focus on an outcome or a phenotype. This can also apply to exposures and covariates. But for simplicity and to keep us focused, we will we'll keep our swim lanes around that. So for a moment, can you guys still hear me? Because I'm getting stuck back there. Can you hear me? You can't. Okay, so I have to stay back here. Okay. For a moment, let's bring it to life. Imagine you're sitting at your desk or on your couch or in your remote location, wherever you are, and you have something you're working on. It's a task, it's a project, it's a grant, it's a manuscript. It's a request from a cross-functional team, if you're in pharma. and you have an outcome, and you need to define it. And you know you need to start with the code list. And I think we've all been in that position, or maybe we've been in situations. What do you do next? And we can imagine for a moment, we Google. We can imagine we go to resources and repositories. We maybe go to other publications. So there's a myriad of matrixing in on how we can think about developing that code list. And that's where we notice the heterogeneity. And for me personally, there's been lots of heterogeneity in how code lists are developed in my career. And again, it depends on the use. What are you using it for? You can't spend 2 months developing a code list if you need it for a quick analysis in some secondary data to see if you have enough cases. It also, and we'll be speaking about this today, then we'll talk about it, your outcome. Is it an acute outcome? Is it a more broad outcome? So those are some other considerations. But again, where do you go next? Who do you collaborate with? Are you doing it by yourself? Or do you have a function within your organization that helps with coding? Most groups, or a lot of groups, don't. Are you looking to a research assistant? So who is collaborating here? Are you bringing in your clinicians? Are you bringing in maybe the data analysts that actually know the data? So these are some of the things, thinking about it, how we should and consider moving forward to make sure that we right up front get the quote-unquote code list right. And I think this will come to life when we see the presentations today. And we will see how the upstream leads to the downstream. And then finally, how you disseminate it. How do you put it in the appendix of a paper or the appendix of a grant? Or maybe it's a footnote in a small table that you've created so that you can essentially memorialize it so you know exactly what you're speaking about. And so without further ado, I would like to present our first speaker, Sina, and take it away.
Thank you. All right. I have to adjust this because I'm short. Thank you, Susan. I hope everyone can hear me okay. Okay. So my plan for the next 10 minutes or so is to review with you how in the service industry we typically develop code lists for phenotypes for use in the studies in real world that include codes. The steps that I'm gonna, before that, here are my disclosures. Okay, let's jump right into it. The steps outlined here is just a proposed way of thinking about the process. These developed code lists form the building blocks of algorithms, which are then used to clearly define computable phenotypes. And when I say phenotypes, I mean be it exposure, covariate, or outcome, or any other variable that we typically think in the context of a real-world study. In the first step, we often begin by defining our phenotype. The main goal in this step is to come up with a definition with as much details as possible. We want to, at least from a clinical standpoint, have a crystal clear sense of what we are trying to measure. However, we also have a challenge. A lot of code systems or vocabularies, such as International Classification of Diseases, ICD-10, often do not contain the level of granularity and the specificity that we are looking to. This is especially the case a lot of times we are interested in phenotypes that are due to a certain etiology, as is the case here with the case of hyperglycemia, where we are not only interested in hyperglycemia, rather hyperglycemia caused by intake of glucose steroids in a context of a certain autoimmune disease. To quickly showcase this challenge, let's flash forward to the fourth step, developing the code list, and look at some ICD-10 codes together. First, we have a T code that, while captures the adverse effects of corticosteroids, it fails to distinguish between hyperglycemia versus other types of adverse events. Then we have the E code, which does get at hyperglycemia, but has two issues. First, it doesn't specify corticosteroids, but also usually gets at the most severe cases of this adverse effects when hyperglycemia progresses to full onset diabetes. So with this code, we're gonna only capture the most severe cases. And finally, we have the unspecific hyperglycemia code, which we can then use to assess in a population who of this particular immune disease who just started taking corticosteroids and look at the incidence of hyperglycemia, which again cannot distinguish between the different etiologies causing hyperglycemia. I just wanted to quickly highlight this example to make the case that while often we need to have a clear sense of the clinical concept, the phenotype that we want to define, we have to balance that against the level of granularity and limitations of the code systems. And when we have to make decisions, and it often involves actually getting creative. I picked just one aspect of defining the phenotype. We can look at it from a number of different angles. Other presenters will look at it from a different lens. So let's move on to the second step. In this step, we want to determine, as Susan mentioned, the level of validity needed for defining our phenotypes. At what end of the spectrum we have the feasibility analyses that are not intended for publication, they're for hypothesis generation to inform a future and upcoming study. At the other end are regulatory submissions using real-world data. And there is everything in between, and the majority of the work that we do is actually the everything in between. Sorry, I should have advanced the slide. And we don't have clear guidelines as to the level of the validity needed for what is the majority of the work, the majority of the studies that we do. What we do have at present is the US FDA's guidance recommendation for the use of EHR and claims in regulatory communications. The draft guidance recommends that operational definitions in the study for phenotypes, be it exposure, outcome, or covariates, to be validated against a gold standard, and the validation process be documented, a strength and limitations be discussed. While this might be relevant for regulatory purposes, this threshold set forth by the FDA may not necessarily be applicable to the majority of real-world studies that I mentioned that we typically do in the service industry and even in academic setting. Let's move on to the third step. The third step involves doing research and reviewing the body of existing literature. I want to quickly go over 4 of these. First and foremost, of course, we want to look at the published literature, peer-reviewed publication, not only original work on the topic, but also the methods paper, and those that involve assessing the methods for validating the phenotypes of interest. Moving on to the second resource, there are public list repositories that are often really useful. There are so many of them. I decided to just highlight a couple of them. One is the COVID-19 Sentinel program. When the COVID pandemic began to support efforts for studies in real-world data on COVID, there was a master protocol put in place under the leadership of Dr. Lorey, and this contains a rich list of code lists for a number of conditions that have been peer-reviewed and QC'd that go beyond just COVID-19 and capture a lot of other phenotypes that we typically study in our real-world studies. This effort also leveraged the work that has been done in the CMS chronic disease data warehouse, which also contains code lists for a list of about 30 chronic diseases that are often covariates of interest in a lot of our studies. The third resource that I want to highlight are the guidance documents by the payers, especially by the CMS, that are developed and include often detailed code lists for diagnoses, procedures, and lab codes to aid in the reimbursement of administrative claims. What has become more and more common in the past few years are also guidance documents by the biotechnology companies that have the detailed instructions with codes to help processing claims related to their approved medicines and devices, and that can also be a really helpful resource in studies involving those. And finally, the final resource is leveraging the data itself, which can be done in a number of different ways. One way that I find really helpful is within the context of the population of interest, look at the most common diagnoses, procedures, lab codes, medication codes, etc., and sort of compare that against the code list that we have already come up with using the resources that I mentioned before. Oftentimes, the data itself points us to directions that we weren't considering and initially thought that would be out of scope for the code list that we are trying to develop. The fourth step is where everything comes together to form code lists and, as needed, algorithms. Considerations in this step involves mapping and crosswalk between different vocabularies if our initial code list has been developed with one vocabulary and needs to be expanded to a prior version, for example, ICD-10 to ICD-9 and vice versa. Additionally, at this step, I think it's often useful to think outside the box and go beyond just one code system and vocabulary. The aim of this is to capture as many patients as possible. This is particularly important for a lot of studies when we have a short look-back period, a baseline period, to capture all the comorbidities. For example, imagine a study that we have diabetes is a comorbidity of interest, and we only have a 6-month look-back period. Our experience suggests that if we only leverage the diagnosis codes, we're going to miss a lot of patients. And that's because in patients, especially with long-established diabetes, those codes are not likely to show up as often. One way to overcome this would be to expanding the code list and, for example, include LOIN codes, and if the lab results are available, consider NDCs, HCPCS codes for medications, and even look at the CPT and HCPCS codes for procedures that are exclusive to patients with diabetes. Ideally, at this step, we want the code list to be reviewed by clinicians, medical coding expert, and also data analysts. Often data analysts have unique insights because they are so much closer to the data and have a better sense of how these codes are populated and used in the data than us pharmacoepidemiologists. And then the final step is to pressure test or develop code lists and algorithms in the database of interest itself. One way that we can conduct this is to take the estimates generated using our code list and then benchmark that against a reliable population-based estimate. Significant deviations between the estimates generated between the two might flag some issues with our code list. However, I should note that this process is not equal to validation because we might have perfect agreement between the two, and yet our algorithm be not valid and have a positive predictive value of 0. So this was a quick overview of the process. Before concluding, I want to highlight a few key messages. First, the process of developing code lists in the real world is often deliberative and time-consuming. We often don't budget for this in our study timelines, and that will negatively impact both internal and external validity of our study. Second, I presented this process as rather straightforward and linear. That's often not the case. We often go back and forth when the new information becomes available, and it actually gets quite messy. And finally, a point that Susan also mentioned, while code lists and algorithms are really important in studies involving real-world data that have codes, we often receive little to no training, both in academia and in the service industry, on the methodology and best practices on developing code lists and algorithms. And with that, Thank you so much for your attention. I'll turn it over back to Susan.
Thank you, Sina. And that is really to help us set the stage. Our next speaker, Dr. Loray, thank you.
Thank you. Thank you, Susan. And certainly thank the organizers for selecting this for presentation. So Sina talked about developing the code lists. I'm going to focus specifically on validation. Sina, which one was
So
it?
10:30.
10:30, got it. Got
And
it.
then,
Thank you so much.
Of course.
Wonderful.
Great. And then we.
Yep, got it.
Okay, perfect.
So I'm Vin Loray. I'm at the University of Pennsylvania. We heard about the development of the code list, the steps. I want to talk about validation studies of algorithms for health outcomes, just to give you my disclosures on this slide. So I'm going to first give you the rationale for validation of algorithms, or as Sina talked about, computable phenotypes for health outcomes of interest in real-world data. I'll then talk about the conventional approach to validation studies for outcomes. I'll talk a little bit about validation to inform quantitative bias analysis, and then briefly conclude with alternative approaches to ascertaining health outcomes. Okay? So identification of health outcomes in real-world data using classification algorithms may be inaccurate. The database studies that we're conducting, analyzing using real-world data, were not designed for the research that we necessarily intend. And the algorithms may be inaccurate, such that patients with a disease, cases, may be classified by the algorithm. Patients without the disease may be classified by the algorithm as having the disease. Those are false positive errors. Alternatively, patients with the disease may be classified by the algorithm as not having the disease. Those are false negative errors. And there's a number of different factors that may affect the accuracy of algorithms in real-world data. So you see at left the setting of the diagnosis, whether it's in the outpatient setting or in the hospital setting. Perhaps in the hospital setting, the clinicians have more time to confirm diagnoses, to record the discharge diagnoses compared to the outpatient setting. Sina mentioned the lack of granularity of codes. Just as an alternative example, there's codes for example, toxic hepatitis. If you're interested in drug-induced liver injury, that may not necessarily be specific for that condition. A single diagnosis, particularly in the outpatient setting, may be a diagnosis that a clinician suspects, but on a subsequent outpatient visit may not be confirmed. May be a rule-out diagnosis. You may have improper use for reimbursement by clinicians who aren't honest, and then there may be issues with missing data and data errors. And these algorithm inaccuracies can lead to, in the parent study, outcome misclassification that could ultimately lead to biased estimates of treatment effects. So validation studies are important to pharmacoepidemiology, to understanding outcome misclassification with our case-finding algorithms. Validation studies can evaluate the accuracy of the algorithm compared to some reference standard. They help us understand and assess the degree of algorithm error and the potential for misclassification. And as you heard from Sina, they're often requested for studies submitted to regulators prior to the conduct of the main study. But unfortunately, researchers receive fairly little formal training often in validation. So I'd like to take you through the conventional approach to algorithm validation to sort of give you a step-by-step approach. So it starts obviously with selecting the health outcome of interest. And I just want to pay a little attention to the selection of this outcome because pharmacoepidemiologists can select acute, severe events that have a clear date of onset where the patient will present for medical care often imminently, or a chronic indolent event which may have an unclear onset and may not prompt presentation for care. So for example, I spend a lot of time studying liver diseases. End-stage liver disease, decompensated cirrhosis, is a very clinically overt outcome, an acute severe event. It's characterized by variceal hemorrhage. Somebody's bleeding out of their mouth, their belly fills up with fluid, ascites, they're confused, encephalopathy. This is clinically overt. Patients will present imminently to the hospital. In contrast, compensated cirrhosis, patients can exist for years without ultimately presenting, and so may not prompt presentation for care and may be very challenging to determine the exact date of onset. So considering which outcome you're going to evaluate is an important first step. Then devising the case algorithm to ascertain that health outcome of interest. You heard from Sina, it could be based on code lists, often diagnoses alone, procedures, medications, or some combination, but you could include other variables like laboratory results, the specialty of interest, whether it's rheumatology, infectious diseases, or the place of service, outpatient or hospital. Important preparatory work prior to development of a case-finding algorithm. Review the literature. What were the data? Are there any published algorithms? Have they been assessed for accuracy? Work with your consulting clinicians. Determine what are the symptoms that classify this health outcome. How is it diagnosed in practice? What kind of therapies are used? It's important for you to be able to determine what kind of an algorithm and what are the components of the algorithm that might want to be included. Consulting your database experts to know what variables are present and included in the data, and what are the practice patterns and whether there are any variability in the practice patterns. A number of options available for algorithms to identify health outcomes. Many algorithms in pharmacoepidemiology use diagnoses alone, often hospital discharge diagnoses. If you're going to use hospital discharge diagnoses, you have to consider, are they going to be principal or primary diagnoses alone, or would you be interested in contributory or principal, primary, in essence, in any position? Are you interested in potentially using outpatient diagnoses? Should it be one outpatient diagnosis? Or are you worried about rule-out diagnoses that are suspected but not confirmed, in which case you want to maybe use two or more diagnoses? Maybe you want to use laboratory-based abnormalities. Maybe you want to use a combination of diagnoses plus labs or diagnoses plus drugs. For example, if you want to look at TB and you wanted a diagnosis of TB plus anti-tubercular drugs. Many, many options to choose. So after you've developed your case-finding algorithm, then the next step is to determine the reference standard. And predominantly in pharmacoepidemiology, the reference standard is the medical record. But in some cases, it may be a disease registry, or it may be, for example, a questionnaire administered to a general practitioner. Really depends on the question and the kind of outcome and the availability of the data. Once you've developed your case-finding algorithm, determine what the reference standard is, then, particularly for those who are going to go into medical records, formulating some definition for that outcome, typically based on clinical review, looking at guidelines, who your clinician, consulting your clinicians to determine how are they practicing, what are the standards that they're using, consulting your data experts and patients as well to formulate that definition so that clinicians who adjudicate the endpoint will have a systematic approach to apply the definition to when they evaluate their structured forms. Just to give you some example of a definition that we have used in our own validation studies, we did a study to validate decompensated cirrhosis, hepatic decompensation in our data. We defined decompensated cirrhosis based on any of the clinical complications of cirrhosis, ascites, peritonitis, variceal bleed, or encephalopathy. We came up with definitions for each of these diagnoses and classified an event as occurring or not if decompensation met any of the definitions for any of the diagnoses consistent with decompensation. So once you've come up with your case-based algorithm, you've determined what your reference standard is, and you have come up with your definition, then you have to select the sample for validation and collect the data to classify that outcome. Typically, sample size and validation is based on the desired precision around the estimate of the parameter of interest. So for example, if you were interested in a positive predictive value of at least 80% and wanted around plus or minus 10%, 95% confidence interval, that may be around 75 patients selected for validation. For those who are then going to go into medical records, you have to determine what components of the medical record to review. Are you going to go into admission notes, specialist notes, discharge summaries? Are you going to use radiographic reports? Are you going to use pathology reports? And then work to create structured data abstraction forms for abstractors to place the data onto. You have to train the abstractors and then pilot the abstraction to confirm the reliability of the abstraction process. And then you adjudicate the health outcome. That may be determining the agreement with a disease registry or a general practitioner questionnaire, or it may be expert review of structured forms to confirm that outcome. If you're using expert review, the adjudication usually involves one or more clinical experts who will review these data forms, who will then use the outcome to adjudicate, is it present or is it absent? What we've done in our group is to use typically 2 clinical experts who independently review the structured forms, apply the definition that was developed to adjudicate was the outcome present or absent. And if they disagreed, then adjudication would commence with the 3rd reviewer to act as a tiebreaker. And then lastly, you determine the algorithm accuracy. And typically in pharmacoepidemiology, the focus is on the positive predictive value because if that's high, that gives clinicians and researchers confidence that outcomes are true events. And typically, PPVs of 80% or higher are considered fit for purpose. PPVs less than 80%, the concern is that there may be just too much misclassification. And so you heard from Sina a bit about refinement. You may want to identify, are there poor performing codes, for example, that may be removed, and you may want to resample and reperform the outcome validation. Now recognize that that's specifically focused on false positives. In some instances, you may be interested in false negatives, and in those instances, you may want to take a sample of individuals who have not met the algorithm and have the adjudicators review, was the disease present or not. Recognize that the algorithm accuracy may differ by data source, and the algorithm that is validated in one data source may not necessarily be transportable to another. Differences in prevalence, differences in reimbursement, and the ways that the disease is managed. And in those instances, revalidating the algorithm in that database of interest would be warranted. But this conventional validation approach may not necessarily avoid or minimize outcome misclassification. And simply getting a single overall PPV may mask differences in PPVs between your exposure groups of interest. And if the positive predictive value or the sensitivity differ in your exposure groups of interest, ultimately your outcome misclassification can be quite large. There are statistical methods that have been used to correct for outcome misclassification. One of them uses only positive predictive value by Brenner, but that assumes that there is non-differential sensitivity, which may not be true. The ISPI database SIG several years ago, led by Gillian Hall and Rosa Gini, developed an app that adjusts the estimate of the measure of effect using a plausible range of positive predictive values and sensitivity in the exposure groups. So this is an alternative way to correct for outcome misclassification. And then lastly, there was a recent great commentary by Lanes and Beechler and PDS that was advocating to stratify validation within exposure groups to inform quantitative bias analyses and to measure sensitivity and specificity in each of these exposure groups, though they acknowledge that this may be challenging in particularly rare outcomes. And I'll just conclude with the field is moving such that there are alternative approaches to ascertaining health outcomes in real-world data. There are certainly, we're seeing machine learning, natural language processing approaches that are incorporating unstructured, qualitative data from EMR, path reports, operative reports, radiology readings, progress notes to ascertain the health outcome, either using natural language processing alone or in combination with other administrative data. And there have been regression model-based probabilistic phenotype approaches that incorporate demographic variables, clinical variables, to come up with predicted probabilities of being an outcome where you're setting the threshold to define cases. And this was something that colleagues of mine at Penn used to develop probabilistic outcome definitions for end-stage liver disease. So in summary, I've given you some sense. Validation assesses the potential for outcome misclassification. I've gone through a conventional approach to validating algorithms that focuses on PPV. I've discussed some methods to correct for outcome misclassification. I've discussed design approaches to validation to inform quantitative bias analysis, and I concluded with just some alternative approaches to identify health outcomes. And I thank you for your attention.
Thank you, Ben. So now that was a really nice segue into, you can see how the actual coding list, the development, really ties into the next development of algorithm and validation. And Dr. Golazar will give us a really nice use case and more details around continuing this, this process of outcome phenotype and validation and algorithm. Asia, thanks.
Hi, everyone. Let me figure out first where I should go. Okay. Great. All right. So, hi again. Very happy to see a lot of old and new friends here. So I'm just gonna start talking about reproducible and scalable phenotype development. Nothing to disclose. But before getting into a little bit of the meat, I want to talk about a real example. We've been working on looking at the risk of stroke in patients with atrial fibrillations who are new users of rivaroxaban comparing it with new users of apixaban. And the idea was to look at it across a network of observational databases, not one, many. So starting with putting the design together, the first thing that we wanted is a definition of stroke, right? What are the things that we need to consider? Are there any valid definition that we can actually start looking at? And what are the issues? What are the things that we have to think about? So just looking at the literature, there's actually a lot of validated literature on stroke. Not one, many, which is great. It's a great start. But they don't seem to agree. There's some differences, and then going further into the detail, well, they're not necessarily done in the same country, same database. Setting is different. Some are looking into ambulatory settings, some into hospitals. Different ICD versions, some ICD Germany, some just like in the US. The position of the code, some of them are using primary positions, some primary and secondaries, some anything. There is no distinction. And there's some nuances. Do we exclude TIA? Do we include TIA? Like the set of codes that we want to use to look into stroke, they're very different. So that creates a whole challenge. Where do we start and what should we do? It's all stroke as an outcome. And I think we're all here because we all agree that we need phenotypes And the reason we need that is that the problem is in the data. All we see and what we use is a snapshot of patient journey that we get from the different sources. It can be EHR, it can be claims, sometimes registry, sometimes the linkage, right? So everything that we see in a patient's journey, in the patient history, like from conditions, drug procedures, measurements, pretty much part of the continuum of care that patients are getting, they are being collected and they're in the records because of, for the purpose of medical care. And a lot of times they represent an evolving process, what the patient went through to get the proper treatment. And it's incomplete. We only see a piece of patient journey. And going from one location to the other, one setting to the other, there are always differences, right? Like what are the recommendations, what's being reimbursed, what's the practice pattern, all of that has an impact in this picture that we see. That makes the phenotyping extremely challenging, right? We know that, like we've heard a lot, the examples show that a lot of time a diagnosis code is not enough. There is a lot of uncertainty around diagnosis code, and usually we need to boost the performance of our phenotype, how we define stroke or any other outcomes or any other phenotype, by looking at additional data elements. So there are a lot of, for example, literature out there. This is just one very good example showing that using multiple elements of an EHR data will give you a more consistent and a higher performance in defining your phenotype as opposed to just using condition codes, right? So that leaves us with a pretty much complicated scenario, right? So we need to have an in-depth understanding of the disease, how it's being presented, how it's being diagnosed, managed, and the general prognosis of the disease, to get you an understanding of like what the patient is going through and how I can take advantage of these diagnosis code and condition records and lab and anything else within the patient history to start constructing, coming with a phenotype algorithm and defining my patients. And again, that means that like I also have to have a familiarity with medical practices, especially when we're thinking about going more than like one database and one setting, familiarity across different settings and different databases. And the question is that, okay, how should we do that, right? So we're all here because we are passionate about generating reliable evidence from observational data, data that is imperfect, has its own nuances, but it has a lot of value. And for this, I think we all agree that we need a reproducible and scalable process for phenotyping. Before I get into some sort of a solution space, I just want to have like a quick intro about Odyssey. I know a lot of you guys know about it, not going to spend a lot of time on it, but Odyssey is an open source community, open source development and science. A lot of passionate folks from industry, academia, government, healthcare system. Everyone, over 3,000 people who are all passionate about improving healthcare. And we have collaborators from across the world, over 3,000, 80 countries, 21 time zone, and 6 continents. There are over 400 databases in OMAP, from EHR registries, claims, administrative claims, different countries, encompassing over 900 million unique patient records. So what's important about Odyssey is that the community, the group of people are all focused on developing standard representation of data from different sources, developing standard solution in terms of analytics to ensure clinical evidence generation that is highly reliable and valid across the different databases in different settings. One of the other initiatives that I want to briefly talk about is Darwin EU. Not going to spend any time on it because we don't have time, and there are workshops and a lot of presentations over Darwin, but Darwin is a consortium, and think about it like as a surveillance system set in the EU. Think about Sentinel, much bigger than Sentinel across 24 EU member states. And we're thinking about generating a sort of like an initiative that can do evidence generation at that scale across different countries and different settings that requires standards. OMAP, a common data model, and standardized analytics are being used in the case of Darwin. This is a 5-year initiative initially. that you will hear more about it in the next sessions. So both Odyssey and Darwin are working on basically using the same principles of following through a journey of to generate reliable evidence, and this is based on standardization. So if you think about it, we have standardized data that is coming from different sources. Combining it with standardized analytics, you can answer a lot of different questions. How are patients being treated? What's the incidence rate of my outcome? Stroke in my example. Are apixaban users experiencing more stroke compared to rivaroxaban users? Using different design in principle, and also looking at individual patients. Is my patient at higher risk of stroke who's an apixaban users? And what is the risk of stroke in 1 year after use? So all of that, the core principle and foundation of all that is having phenotypes, reliable phenotype that can be used to answer all of these different questions. And since many, many years ago, so there's a lot of activities within the community focusing on developing methodologies that can enable and ensure definition, development of reliable phenotypes. Both also Darwin is also specifically working on that, and I'm going to just talk about one of the approaches and highlight some of the work that's been going on within the community. As we've heard from everyone, we always start with defining a clinical idea that we want to phenotype. I want to look into stroke. We divide the process into the two different stages, defining the design, coming up with a systematic approach to define the design. First, identify what is it that we want to phenotype and what are the specific elements that we need to know to decide what codes to include and how to build our phenotype. Once the core components are defined, we use that to define the core components. When do we start looking into the patient? What is the index state criterion? How are we going to exclude and include specific patients using, for example, measurements or different lab values or different procedures? And then we specify the components, the code list. Which set of codes do we want to use based on those specifications? Do we want to go broad or do we want to go very narrow, similar to what Sina talked about? This combination will give us we can actually come up with a phenotype definition that needs to go through evaluation. So there are two different evaluation processes where we look into the descriptive statistics that gives us an insight into the performance of the cohort. We call it cohort diagnostics. And then we also use methodologies that look at empirically evaluate the phenotypes using predictive modeling. And the process is iterative. So the way we start, this is one example, we go through a checklist to identify the core pieces, the pieces that we need to know to design our phenotype. Just giving you an example. Awful lot of times, stroke is a very good example. Patients are being diagnosed or the code is being used in a patient's medical history. They're not diagnosed yet or they're in the process of being diagnosed. Sometimes this is happening because the patient has to get the appropriate medical care for the diagnosis to happen. So we see it in the case of MI or stroke. What we need to do, I need to identify stroke patients, not those who have gone through that. So we have to come up with very specific definition to pretty much account for this overcoding problem. Similarly, that can happen that of a lot of times you see less amount of coding or undercoding of specific conditions. That has an implication of how we can identify these patients in the coding. And for each of them, we use a series of guiding principles to identify how we can actually account for that. Should we use repeated diagnosis? Should we actually require an adequate visit? Should we actually exclude folks that had differential diagnosis after the first while including repeated diagnosis? So the structure process makes you go through a series of these questions to help you identify the elements. And this list goes on and on. Do we want to look into an incident condition or a prevalent condition? What are these elements that we should think about if the condition is incident or prevalence? And it continues on and on and on. Once we have that, this systematic process, we come up with our phenotype definition. We put it together, and we go through a set of diagnostics. Just briefly describe cohort diagnostics that gives us a very comprehensive overview of the patient's characteristics and gives us like an idea of how the cohorts are performing. Are we actually seeing what we're supposed to see? And also at the codes, like, do we see the magnitude of it? The disease is exactly what we expect to see or not? The incidence, are there changes in the incidence of the disease across like different time frames? Is it expected or not? Are we getting enough insight to actually go back and fix? So a lot of that, a lot of work would be, time would be spent on core diagnostics, and based on that results, we go up and we do assessment and alignment per use case and per our need. So this process is iterative until we get to a point that we feel comfortable and confident that we can use it, and this is database specific. So diagnostics and the potential use of any of these phenotypes are per database, and it never means that we're like, as long as we say this is good, it's going to be good forever. So just to summarize, just going to repeat it, it's a challenging work to define phenotype. And we've heard about it, we've seen the literature, we are all trying to do a good job, but the good job is not enough. And as we see it, we're seeing not repeatable. We're all following the literature, consulting with experts, coming up with a process, but unfortunately they're not the same. We see different definitions across the literature, and that creates a problem. Which one do I trust? If all the definitions are correct and they don't have an impact on the findings, that creates a question of like, why are we going through this process? I think we all believe that it's important to have the correct definition. So we have to account for that heterogeneity that we see, and we have to come up with strategies and approaches to minimize that. And I think as a community, as a group of friends and collaborators and colleagues that are sitting here, we all have to work together to come up with standards, a process and structured approach to be able to have phenotypes that are actually repeatable. We can trust them, phenotypes that can be used to ensure reproducibility. Thank you very much.
Thank you, Asya. Yeah, it's interesting how we're seeing the development of this symposium today. We started out with coding lists, which Sina mentioned are iterative, through the validation and the algorithm development, which is also iterative. And now we're talking about phenotypic definition and how qualitatively, as we're defining this, is also iterative. So it's a nice journey that we're on. And our next speaker, Dr. Ko, will be giving us the industry lens perspective on all of this. Thanks, Doug.
Thank you. Good morning. Let's see, how do I? Let's try this again. There we go. All right. So, good morning. My name is Doug Kuo. I'm the head of epidemiology and risk management operation at Beijing. So I'm very happy to have this opportunity to present to share some experience from the industry perspective. And I have to admit, it's extremely difficult to follow the previous speakers. They definitely have laid out a pretty high bar for me to keep up. I hope I don't disappoint. So my goal for the next 10 minutes is really to walk you through a scenario on how to propose a study for the purpose of meeting a health authority required safety monitoring study, and try to incorporate some of the concept and some of the challenges. But as previous speakers have already elaborated on, my goal is really to come up with a range of considerations and topic area for you to think about, but it does not mean I have the solution to all those questions. So look forward to the discussion at the end of the conversation and see maybe everyone can brainstorm a new way of doing things. So here are my disclosures. So as an epidemiologist working in the industry setting, right, especially in a pharmaceutical company, so here are some unique areas where pharmacoepi plays an important role, either in the pre-approval space when you try to bring epidemiology insight, or more specific from the safety perspective to support or enable decision-making around the clinical development program at different stages. But once the drug is submitted for drug approval, then also there are unique opportunities to provide some insight or to bring some contextual or supported evidence to facilitate negotiation with the agency, or also to support your clinical summary of safety or efficacy endpoints. And obviously in the post-approval space, there are also a large variety of activity that you can do as well. So if you think about some of the concept around developing a robust, meaningful coding list or algorithm to identify the outcome or the exposure, we'll have the right phenotype. So I'm going to add a new element to this, speed. So if you're in the pre-approval space, you can, based on literature or prior knowledge, and identify certain outcomes or exposure that may require some coding algorithm development or even invest in validation effort. But if you're in the peri-approval process, in an active interaction with health authority or agency during review process, there may be a new health outcome that came up that was out of your proactive planning list. Now you have a short period of time to come up with a robust way to generate data to support or to explain your safety signal. Then in the post-approval space, usually most of the post-approval study are pre-specified, negotiated, and agreed with the health authority. And it is extremely difficult to modify the study protocol once it's endorsed or approved. But with the rapid evolution of use of real-world evidence, new data platforms, so your study is actually in a very living and dynamic environment. The protocol that you proposed at the time was adequate or robust, but in 5 years, there might be new data sources, new registries, new way of collecting data, so you all have to coexist in that environment as well. And I think this is one aspect that many of us fail to appreciate, is that a study that was agreed with the health authority, but 5 years later, it may look like, why did they make this decision? Why they haven't updated their approach? So we have to anchor around the health authority agreement, endorsement, but at the end of the day, as a sponsor of this drug, we have to provide periodic benefit risk evaluation report to health authority. There's one key component around literature review. So during a particular reporting period, you have to disclose what type of literature review you are conducting, and you have to summarize any new finding you find in those literature review. And you have to incorporate that with the ongoing study that you are currently leading and come up with a most up-to-date benefit risk evaluation. So beyond all that, if you calculate backwards, evolution of code lists, evolution of new data sources, evolution of health authority thinking around code lists and algorithm development, you have to put all that into consideration as you come up with a general conclusion in your periodic safety report. So that's the public health obligation from the sponsor to the agency and also in a very crowded space. So I'm going to break that into different pieces, and hopefully at the end there we can stimulate some great discussions. And as Donna Rivera have eloquently elaborated on during the plenary session, FDA had come up with large array of guidance documents, and that really captured the evolution of their thinking around the use of real-world evidence. There is so much emphasis on fit for use, fit for purpose. So the algorithm you develop to define based on characteristic may not be robust enough to use to describe outcome, right? But then if you say, okay, if you have 30 baseline characteristic you want to develop and to study in your study, does that mean you need to come up with 30 new algorithms, 30 new code lists? But or do you put all your effort on developing a code list or algorithm for the outcome of interest, which may be much fewer than the list of baseline characteristics you want to include? So then you have to take into account of the specificity, sensitivity, and the positive predictive value, and then the trade-off between the time sensitivity and how soon you need this data. So you need to put all that into consideration. And the other part I learned the hard way is, as FDA evolved in their thinking, they don't always remember your initial interaction might be a year or two ago before the new guidance come out. And all of a sudden you look like you don't know what you're talking about, although that was a guidance document you were following at the time. So as you negotiate a new study, you have to take into account the evolution of the guidance and the regulatory thinking. If a new guidance come out for 6 months, yes, they may have remembered, okay, we just issued a guidance document, this proposal was before that. But in a year later, they may not be so tolerant about your now come across as a Jurassic approach to doing a COLIS. And so you have to take all that into consideration, especially in the peri-approval space when you're actively negotiating a study. And oftentimes, this study can go on for 10 years, 15 years. In rare disease setting, it may go on for 25 years, just because you have to monitor for long-term safety. So that's where the speed and the dimension of time really play a very tricky aspect in all this development process. Additionally, as one of the speakers mentioned in the plenary session, there's the ICH reflection papers. But some of us representing industry also work on the ICH M14 working group. It's supposed to harmonize all the guidance documents to come up with a general framework how to plan and propose the pharmacoepi studies. My involvement was limited until about 2 years ago, and then obviously the draft guidance document will come out very soon. So there will be another guidance document that represents the most current thinking that harmonizes the regulator from across the globe on how to propose a pharmacoepi study. But there are common themes among all the guidance documents. It's around how do you define the target population? What are the key variables that you want to study, and what is the completeness or accuracy information from the data source? So the theme remained the same, fit for purpose. What is fit for purpose, but from two perspectives? Is the data source fit the purpose you want to use it for? Is the algorithm fit the purpose you try to describe the outcome of the target populations? Additionally, their exposures, outcome covariates. So you have to, sometimes if you're short on time, you only have 1 year to propose a study, you have to figure out where do you invest your effort, where do you invest time, if there's no algorithm available. If there's no time to do validation study, then you have to come up with a solution how to work around all that. So some of these concepts have been repeated over time, right? What is the type of data? I think it's super important that, yes, there are many sessions at this conference focused on advanced analytical methods on how to control bias. But from my perspective as a data scientist, I always think about the foundational issue. Where do I get that data from? What is the construct of the data? What sort of healthcare environment that data come from? Because that dictates the generalizability and sort of outcome you can look for. For instance, in my previous life, I was supporting a program looking at patient renal transplants because of end-stage renal disease. They're on the wait list. And if you look at the commercial insurance claims database, you find a certain estimate on what the proportion of patients with end-stage renal disease. But in the United States, end-stage renal disease also eligible criteria for you to receive Medicare services. Therefore, you actually migrate from one healthcare setting to another healthcare setting for up to 3 years, then you migrate back to your own original employment-based healthcare setting. So how do you capture that patient as they migrate from one to the other to do a long-term observational study? That is really something that I sometimes thought maybe as we develop COLIS, maybe we should also have medical anthropologists who are familiar with different healthcare settings on their team, in addition to clinicians, statisticians, clinical development, epipogs, and even SAS programmers, because they know where to find those data in the very large, complex, big data. So this does take a village. And I look around the room here, there are so many colleagues here who are working on the program that I'm about to share as an example on how we go through that journey. And I don't think I need to repeat this part, the importance of validations. But at the same time, there's so much validation that's been done already. And so I want to bring a new concept that hasn't been mentioned in many sessions I attend, is during the drug approval process, FDA has to go through a process called Area Sufficiency Determination. ARIA stands for Active Risk Identification Assessment. It is the agency's position on whether the Sentinel network can evaluate the drug exposure and the certain outcome of interest. In that document, FDA will list all the algorithms they look at, all the data sources evaluate. What is a positive predictive value? What algorithm is appropriate to study outcome or is not appropriate? So before you really start de novo on a codeless algorithm, you can look at what FDA has evaluated from an algorithm perspective as part of a drug approval process and look at what are the codeless they use. It goes both ways, right? So you understand, okay, I don't need to duplicate effort. I can use what FDA is using. But it also means when you propose a pharmacoepi study, you better be very familiar with what FDA have evaluated already. So you don't propose something that's less robust than they have evaluated in the past, because it set the expectation that you are not trying hard enough. So you don't want to have that perception of appearances as well. So like I said, this is a highly fluid and dynamic environment. I never thought data space study could be this exciting, but now you're adding the active, evolution, the guidance document. If you go to informatic conferences, there are data provider out there busy aggregating data, try to figure out how to track one patient migrate from one source to another. So you have to take that all into consideration when you design a study and also figure out what algorithm, what data sources to use. So the example I'm going to use is a program I worked on until about a year ago, and then I have not seen any of my former colleagues in the room that actually work on this program. Maybe if they happen to be here, or look forward to hear the next evolution of the study. So this is a drug that Celgene have produced, or developed, and BMS acquired. And as a former BMS employee, we were asked to further develop this multi-country safety monitoring study. It's for patients with relapsed multiple sclerosis. So this is the FDA approval timeline. There are 2 indications now for multiple sclerosis, but also ulcerative colitis. But what's interesting that prior to the actual FDA approval day, because of the awareness of the drug potential efficacy, we start to capture in real-world data of patients using this drug. For instance, in ulcerative colitis patient, although FDA approved the drug in 2021, but if you look at record after 2020 and up to 2021, you start to see multiple sclerosis patients taking this drug as well. So there are some degree of off-label use. So defining the index day, the study starting period is critical, especially if you use real-world data. And EMA, so in Europe, they also approve over a very similar timeline. So how do you approach this? As part of the negotiation with the European Medicines Agency, we were asked to develop a multi-country database or multi-country safety monitoring study, and they encourage us not to compete with a country-level registry study. So that means they want us to use various real-world data sources to do such study. If you look at the outcome of interest, like Vince elegantly stated, there are acute outcomes, but there are also chronic outcomes. Then there are also outcomes that's extremely difficult to operationalize, such as acute liver injuries. And there are also outcomes that sounds even more difficult, such as opportunistic infection. But then how do you take it to the next level as serious opportunistic infection? How do you operationalize concept into real-world data study? And also, if you look at there's timeline. So these are information publicly available on clinicaltrial.gov. If you look at different outcomes, have different time window, right? So if you think about typical real-world data, the average duration of follow-up. So for instance, in the US insurance claims database, you'll be lucky if you get up to 5 years of follow-up. But if the health authority is asking for 10 years of follow-up, then you have to be very selective on which data source you use. And also, you have to look at, in the European setting, in the UK setting, there are so many real-world data out there, right? And they all capture different type of healthcare environment setting. So in the UK, there's a hospital treatment inside medical record. CPRD has a medical health record database, but they also have a hospital episode statistics. In Germany, there's the statutory health insurance database, and in France, there's SNDS databases. And many of them are not industry friendly, I will just admit to that. And it's difficult to work with these national level databases in Europe. And usually you have to work with academic collaborator in order to analyze the data. And that become a contracting nightmare as well. So I just put that out there as well. If people have a way to work around it, that's even better. Yes, there's the Darwin system. But the last time I tried to interact with Darwin system, they said, well, we're not quite ready for industry interactions yet. So that is not quite there yet. And also in the US, yes, we have the Sentinel system, but the only way to collaborate with the Sentinel system is through the Reagan-Udall Foundation. And that also means there are certain data partner agreement you have to learn to work with. So that is extremely complicated. So if you think about all this, it's around, we do all this activity around the time of PERI approval. In the past, until about 2 years ago, usually if you look at the approval letter, it will say, up to 1 year after drug is approved, you have to propose your final study protocol and initiate the study. But in some of the more recent approval, that 1 year window is gone. Now it's 6 months. So if you, by the time when you figure out how to work through the procurement, how to decide what algorithm, what code list to include your protocol, and to come up with a preliminary list of data partner willing to work with a pharma company, that 1 year, 6 months is not enough. So you almost have to start developing this protocol at risk, maybe a year before the drug is actually approved. And also, there are existing registry studies as well. So how do you incorporate registry study into your multi-country study? That is also a challenge. So without going through the, I know we may be short on time, but even registry study are not all made the same, because different registry study have different level of specificity around clinical report form, or outcome report form. So, and they also have different monitoring schedule as well. So how do you harmonize all that? That is the challenge. So when I will work on this program, we barely survived proposing a multi-country study that EMA was willing to accept. But we kind of kicked the can down the road. Okay, at the end of the study, how are we going to analyze all the study together? Do we do a federated analysis? Do we do a meta-analysis? Or do we find opportunity to pull all the records together? There are multiple technique mechanisms to summarize the data. So these are all the challenges you have to think about much further down the road. And coding algorithm. Right. So if the coding for the purpose of medical record is, you may have different algorithm versus coding or algorithm for insurance claims database. Like Vince, I talked about, you take multiple outpatient records. But if you're a medical record database, you don't need that multiple record, right? Maybe one is enough. But also remember, medical record is designed to help physicians deliver care. So they may not enter the same information over time. So when you design the look-back window, you might have to consider typical insurance claims database, you put 1 year, but for medical record, you may have to go as far back as possible, right? So you have to build that into your algorithms as well. That's all I have, and I can run out of time as well, so perfect. Look forward to the discussion. Thank you.
Thank you, Doug. We have about 15 minutes or so for questions, comments, and I think maybe come up to the mic, please. Thank you.
Hi guys, that was so nice. I have one comment and then two questions. First comment, sensitivity, right? I mean, if you want to estimate incidence and you don't think what the sensitivity of your algorithm is, you have a problem, right? And then of course, if you can assume that it is differential, that it's non-differential, how you test it, we are developing a test, will be presented tomorrow in a poster, so that's something that should be done. But if you are estimating risk difference, you still need to know the sensitivity. And so we are in the conception and VAC4U environment. We are trying to develop things to get there. For instance, there is already published that if you have both PPV and MPV, you can obtain sensitivity and specificity, which is then transportable, which is a good thing. So consider estimated NPV, because that's a good idea, if at all possible. Sometimes it is. So that was the comment and the question. So I am a mathematician. I program a lot across data sources. For me, code lists are an input. I really don't want to know what's in there. But as a programmer, I would like to understand from those who have the experience and expertise to develop them. 2 questions. First, we said that the phenotype itself it's data source dependent, and I completely agree. I mean, you cannot use the same phenotype in one data source and in the other because the underlying data is different. But the code list, is it okay to use the same code list if the coding system is the same, diagnostic coding system? Is it okay, or do you have experience of problems using the same code list? Would you be, so that's the first question. And the second, Sina was saying, or someone was saying about cross-coding systems. So I understand that there is and this is the Odyssey group that know that, there is, if I have an ICD-10 code or an ICD-9 code, there is a single SNOMED code that uniquely corresponds to it. This is my understanding. So would it be okay to create the code list directly in SNOMED? Or are there nuances that you would not capture according to your experience? So if, or equivalently, if you develop in ICD-10 and then translate somehow automatically to ICD-10, what are the possible problems?
Yeah, these are great granular questions. And Sina, I don't want to put you on the spot, but
I'm Rosa Gini from Mars Toscana, sorry.
I know that you run a coding function, and so please start off, and if anyone else wants to chime in around that. It may be that this is an offline and you need more details.
Yeah, thank you. Thank you for that question. I think just from the experience that we've had about the first question of the same exact code list, can we just use it across different databases, real-world databases, that our experience has been it generates different results. And that has to do with the unique features of each healthcare system, how the data is organized. And so I think what Asia's presentation alluded to, there are a lot of nuances in between different databases, even in the same country in the US with the fragmental healthcare system, that even the same code list might not perform the way we expect it to perform across different databases. And we need to take that into account and develop our code list knowing the limitations and features of the database that we are planning to design our study in. For the topic of essentially mapping, I'll turn it over to Asia. Them come out here.
It's good. Okay, yeah, this works. Rosa, to your second question about SNOMED, I'll answer that first. Theoretically, if the mappings are done and it passes the quality check, a SNOMED code and a concept set, a set of concept, or what is called here a code list, can be used across different databases, right? Assuming that you know the database. So it doesn't guarantee that. Everything needs to be actually worked out in collaboration and with the knowledge of databases and the nuances. I'll give you an example. There's a difference between ICD-9 and ICD-10. I can't remember which one in terms of heavy menstrual bleeding. So one of them has like a code that's called frequent or heavy menstrual bleeding. Right? That's a question. SNOMED has them separately. It's the difference between frequent and heavy menstrual bleeding, right? So when there are nuances like this, there are ways to do that, right? So the SNOMED that has been used for mapping might not correctly capture that. For your own study, if it is a database specific and you know that you can always use non-standard to accompany that. We don't recommend using non-standard, not because it's bad, it is because we want to do network studies. So if I add non-standard to my set of concept code, then the moment I get my concept sets or code lists, and I want to, for example, a concept set and code list that's been used, like using administrative claim data in the US, and I want to use it in the UK, it's not going to work, right? So those are the nuances to know about. There are instances like that, and one of the biggest pretty much amount of time that we spend is to check the concept set that should be included, to look and see what are the other non-standards that we have to think about. The part of cohort diagnostics has a lot of emphasis on the concept set, the things that we don't see that, oh, like the coding system is different and like the mapping has been different because of that. That's something to be considered. But the idea of using standard is, yes, it should technically help as long as the mappings are, like, we have a good understanding of the mapping. So I do phenotyping. I know a lot of people do phenotyping, and we spend a lot of time on the codes and what goes into the concept set to create the phenotype.
I'll just jump in real quick about the SNOMED code. So back in ISB in Montreal, there was actually a poster presentation on using SNOMED code as a starting point to define extremely rare pediatric conditions. So if your data source allow you to use SNOMED code, because obviously SNOMED to ICD-9, ICD-10 is never one-to-one match. So sometimes the condition you are studying could be lumped together with other 220 or 30 disease signs and symptoms. If your data source allow you to identify the SNOMED code, you can actually use that in the extremely rare disease settings as well. So there was actually a poster presentation on that, so thank you. Yeah.
So why don't we, I know we have a number of questions, so this is great. Leo?
Sure, I'll be brief. Leo Russo from Pfizer. So I think this question might be for Dr. Golazar because of your connection to Odyssey, and that is, what about machine learning to generate phenotype algorithms when maybe in the scenario one doesn't exist, or maybe there's one and you want it to be better? Because I heard what you were talking about bringing different things besides the disease codes in there. And it seemed to me that maybe somebody's working on this to take the classification power of machine learning to say, okay, wait a minute, let's really take an exploratory look and say, what is it that really classifies the best? So
Yeah.
I see you smiling. Hopefully that means somebody's working on it.
No, that's actually a very good question. So I mean, it was supposed to be 10, 12 minutes. I don't know how much more I talked. It's very hard to combine everything and talk about everything that's been going on, like especially with phenotyping in the community. And there's one person behind you who's actually very active in phenotyping. But I specifically focused on sort of like a rule-based algorithm and what we go through that, right? So there's a lot of effort on doing probabilistic algorithm, and there are focus. It's not something new. There's Aphrodite, and like there's a lot of work that's being doing. We also use machine learning for prediction. I briefly mentioned that. I don't know whether Joel is here. He's developed a p-evaluator that we constantly use to evaluate the performance, but it is there. Happy to discuss more about it. It was just lack of time. Yeah.
Thank you. No,
You're
it's good to
welcome.
hear people thinking about it. Thanks a lot.
Thanks, Leo. Next question.
This is more of a comment. I'm Bob Chen. I'm the scientific director for the Bryden Collaboration, and I've kind of been the lead for the vaccine safety world, if you will, and just kind of connect the right hand and the left hand within our world. So going back about 20 years ago, starting with different vaccine safety crises, starting with the wholesale pertussis vaccine, intussusception, narcolepsy, and now myocarditis, TTSS. We realized really it's important to have standardized case definitions to look at these rare serious adverse events. So we developed kind of 3 tiers of case definition, and now we have about 70 adverse events with these standardized clinical case definitions. And they're exposure agnostic, so they're appropriate for vaccine, they're exposure for drug, for anything else. And I would say that was kind of vaccine safety 1.0. We now recognize in this new world that we need to move to vaccine safety 2.0. And so we're creating living laboratories where we're planning to kind of update, evaluate, and improve our case definitions using RWE. So anyways, we look forward to collaborating with you. But those 70 hopefully is a foundation that we could build on without having to reinvent the wheel.
Thank you very much. Next question. We have about 5 minutes left. I think I don't know if the speakers can stay a little bit longer, but please go ahead.
Okay, sure, I'll be brief. So I'm Anna Schultz from LSHTM. So one of the kind of big challenges in reusing code lists or assessing code lists is that they're not kind of consistently reported, particularly not the process by which they're created, and particularly around excluding codes. So we can see the codes in the code list, but not the codes not in the code list and why they're not in it. Do you have any recommendations around how to report code lists and particularly metadata around the exclusion of codes?
Yeah, I know it's something that as a group we thought about, and this I called it dissemination, I think, in my opening, and we see a lot of heterogeneity even in published papers and published work around it, and so I think that is something, raising the awareness around it. I'll ask Vin to comment here.
Yeah, that's a great question. I think that when you're conducting the validation studies to show sort of the world of diagnosis codes that you have considered and maybe not used would be really valuable. And this is where sort of where Rosa Jeannie's question was asking, can you use the same code lists in different settings? When you're looking at a whole host of even potential codes, like, you know, when Asi was talking about stroke, do you consider TIA? Would you consider other atherosclerosis, the cerebrovascular atherosclerosis, sort of including all of those, at least to say, this is what we considered. Perhaps these were poorly performing, or perhaps that they weren't used by clinicians. And so getting sort of a hierarchy of how often are these codes used, that's where collaborating with your clinicians, collaborating with your data experts really comes in to determine what should or should not be used and what is potentially transportable or not. But I think reporting them in these kinds of validation studies is an important initial step.
Thank you, and we have time for one more question, realizing that some folks might need to leave, so please feel free. Go ahead.
Hi, I'm Imana, Senior Consultant at IQVIA. So this might be a loaded question, but given the challenges that had arisen transitioning from ICD-9 codes to ICD-10 codes, and then now knowing that ICD-11 codes have come into effect in 2022, how do you anticipate these same challenges from transitioning from 9 to 10 as we're going from 10 to 11? And how do you recommend us preparing our phenotyping structures for this transition as well?
Yeah, I mean, I can start out here. I actually lived the 9 to 10 transition. And so top line is probably a lot of the same challenges, but I'll open it up to my panel here. Anyone want to comment?
I would say standards. So if you go through standardization, the headache and pain of what is that ICD-9? Oh, what is the ICD-10? How about the 11? That is supposed to be streamlined. You're going to have more validity in what you do, efficiency and quality. But I come from the standard world.
Well, it is time, and I want to be respectful to everyone's additional meetings and get-togethers and lunch that may come after this. Thank you all for attending. It was a wonderful, lively, appreciate it.